Hey guys, so just to summarize what we've kind of learned so far, Mendel talked about genes and he talked about these alternating forms of a gene that we call alleles. And so you can have the allele for the dominant trait or the recessive trait. And so remember, genes have a specific loci. Plural would be, or that's plural, sorry, singular would be locus on a chromosome. Okay, so if this is our chromosome, you're going to have a specific loci on that chromosome. One of these chromosomes you got from mom, one of them you got from dad, maybe one of them gave you a big A, and one of them gave you a little a at that loci. But that codes for a specific trait. And so what Mendel understood was that it's the chromosomes that actually separate and undergo independent assortment that gives us the genetic variation that we see. Okay, so we see this here in our diagram. The law of segregation tells us that the two alleles separate from each other, but remember, those alleles are on the different chromosomes. The law of independent assortment tells us that also these non-homologous chromosomes separate from one another. And so that's what we see occurring during meiosis. That gives us all of these different genetic combinations that you see here. Okay? All right, so in addition to Mendel, another really famous guy in genetics is a man by the name of Thomas Hunt Morgan. And so he's kind of neat in Kentucky because he went to the University of Kentucky. If you go downtown, actually really close to Rupp Arena, there is the Thomas Hunt Morgan House, and it's open for tours and stuff like that. But that is where he lived when he was a student at UK, in the Thomas Hunt Morgan House. And so what he studied was Drosophila melanogaster, and that is the fruit fly. Okay, you all probably know those fruit flies that come around fruits. You see them in the summer. Sometimes get them in your house. They're like little gnatty like things that are super annoying. But that's what he studied. Because they were easy to study, they were easy to kind of keep in a jar and maintain and manipulate. And so in the fruit flies and in many organisms, you have what is referred to as a wild type and a mutant phenotype. So for any trait, most organisms in the population will exhibit the wild type phenotype. So that means pretty much what everybody has. Now, some of them will have an alternative to the wild type, and that's known as the mutant phenotype. So these are a little bit different than a simple dominant and recessive traits. And so instead of writing them as, big A, or big A, little a, we write them as a letter with a superscript. And I'm going to show you some examples of that in just a moment. Okay. So, fruit fly are really big. A lot of AP biology does fruit fly experiments. There's one that's an option to do with AP biology. Of course, we're not in the classroom right now, so we're not doing those anyway. But I personally hate fruit flies because you end up with them all over the classroom. The lunch ladies hate fruit flies because they inevitably gravitate towards the lunchroom. So I choose not to do the fruit flies, but they are a really big component of genetics and studying genetics. Okay, so Mendel did the peas, Thomas Hunt Morgan did the fruit flies. So he was looking in this experiment at the eyes of the fruit flies. So he had this symbol is going to be the female, this symbol is going to be the male. And so he crossed his female that had red eyes with his male that had white eyes. And what he noticed was that all of the offspring had red eyes. Okay? So you might look at that and think, well, red's the dominant trait, right? That's what seems to make sense. But when he crossed these red-eyed offspring, the male and the females, in the F2 generation, what he saw was that 100% of the females had red eyes, but the males were different. They were a 50-50 red-white split. And so that was different than anything that Mendel had studied. He said that there's something else going on here. And so there has to be he thought there was something with the Y chromosome, right? So if you remember from when we looked at the karyotypes, the X chromosome is pretty big. The Y chromosome is quite a bit smaller. Okay, but what the Y chromosome has is it has a gene known as the SRY. It's the sex determining region of the Y. That's what that stands for. Okay, so Y determines the development of male testes. Okay, Y determines the development of male testes. If you don't get a Y, your gonads, which are the precursor to the reproductive organs, those are going to develop into ovaries. If you get that Y, then those gonads are going to develop into testes. So again, here's the X chromosome, here's the Y chromosome. That's under a really high-powered microscope there, a scanning electron microscope there. And so remember, females are XX, males are XY. So if you've got your parents, your dad here with XY, and your females are XX, mom is going to always donate that X. Okay, she's always going to give an X to the offspring. Dad can give an X sperm or a Y sperm, but either way, you could have an X or you could have a Y. It's a 50% chance there in your zygotes, okay, depending on what mom or dad donate. So what Thomas Hunt Morgan said is that this gene must be located on a sex chromosome, the gene that coded for the eye color in the fruit flies. And so any gene that's located on a sex chromosome is referred to as a sex-linked gene, okay? So usually sex-linked genes in humans are on the X chromosome, just because the X chromosome is a larger chromosome and it has a lot more genes than the Y chromosome. So for a sex-linked trait to be expressed, a female has to have 2 copies of the allele, okay? A male only has to have 1 copy of the allele. And so sex-linked recessive disorders are much more common in males than females, okay? So let me show you what I mean here. Let's say we have a recessive disorder. Let's say color blindness. Color blindness is an example of a sex-linked disorder. And so I'm going to use little b for color blindness, big B for normal vision. Okay? So for a woman, she could be X big B, X little b, and be normal. She could be X big B, X big B, and be normal. Okay? She could be X little b, and have color blindness. Okay? That's our woman, our girl. Okay? But for our male, he only has 1 X chromosome. So if he's X big B, he's normal. If he's X little b, he's colorblind. Okay? So whereas a female has a smaller opportunity to be colorblind, the female has to get 2 copies of the defective allele. The male only has to get 1 copy of that recessive allele. Okay? And so it means that sex-linked recessive disorders are going to be much more common in males than in females. Okay? So, this is a Punnett square. This is black, so it's a little hard to see. Sorry about that. Big N equals normal color vision. So when you do these Punnett squares, you have to express the percentages for the sex. Okay? You have to express the percentages for the sex. You can't just say 50% colorblind, 50% normal. You have to tell it for female and you have to tell it for male, because it's going to be different when we're talking about a sex-linked gene. You also have to write the chromosomes in here and use the alleles as exponents or superscripts, so to speak, on the chromosomes. So let's work this Punnett square. Okay? So we've got X big N, X big N, X little n, Y. I'm going to go ahead and set these all up, and you can pause me, and I want you to set them and come up with your answers here, and then come back and check with me in just a moment. So here in these Punnett squares, you should have worked those out. The first one yielded 100% normal male and normal female, so that was the same for male and female. This one here was very different for males and females, though. The females were all normal. The males were 50 normal, 50 colorblind. And then the last one, they were the same there as well. 50% normal, male and female, 50% colorblind, male and female. Okay? So you do see, though, that it's important because in instances like this, it may not always be the same for males and females. Okay? And I think that just works out the Punnett square there for you. So you all have an assignment today where you're going to do some sex-linked practice problems. So go ahead and do those. If you have any questions on them, let me know. And if not, then you all have a great day.